The word exosome is on thousands of products right now. Most of them have no scientific right to use it. Here's why that matters and how to tell the difference. EV nomenclature is one of the most confusing areas in the field, and bad terminology is eroding trust in products that people are actually paying for. My name is Dr. Frederick, and I've been studying extracellular vesicles for nearly a decade. This is the only video you need to understand the different types of nanoparticles that exist. Let's start at the top with extracellular particles, EPs. This is the preferred overarching term for all particles in the nanometer to micron size range. EPs split into two families: vesicular, meaning they have a lipid bilayer membrane, and non-vesicular EPs, or NVEPs, which don't have a lipid bilayer membrane. Some examples include lipoproteins such as HDL or LDL, and different kinds of protein aggregates. NVEPs matter way more than most people think. They can greatly outnumber EVs in some biological samples, and most separation methods co isolate them with EVs. That has serious implications for product purity and concentration claims. Alternatively, extracellular vesicles, EVs, are particles released from cells delimited by a lipid bilayer and incapable of self replication. From a purely scientific standpoint, EV is where the terminology should stop for most products on the market. But the term EV doesn't have a rig to it. In other words, it's not very marketable. So most brands out there will just use the term exosome without scientific backing. The problem? Improper labeling erodes trust, and consumers may not be getting what they're paying for. One heuristic to lock this in: all exosomes are EVs, but not all EVs are exosomes. True EVs break into three subtypes, differentiated by their cellular origin, also known as biogenesis. Remember, EV is an umbrella term. The first type of EVs are exosomes, which originate from internal compartments of the cell, specifically the multivesicular body. They're generally under 200 nanometers, but size alone is not enough to call something an exosome. The second type of EVs are ectosomes, also called microvesicles or microparticles, which bud directly from the plasma membrane. They can overlap in size with exosomes to some extent, which creates real confusion. And the last major type of EVs are apoptotic bodies, which are the simplest. They're fragments from cells that are dying. Beyond EV, there's a growing class of EV like particles produced through artificial manipulation, collectively called EV mimetics. The first type are artificial cell derived vesicles, or ACDVs, and the second type are synthetic vesicles. Now, one important caveat about biogenesis terms like exosome: these should only be used when you can actually demonstrate the subcellular origin for your specific source and condition. These terms are actually discouraged by experts in the field. Also, terms like exosome like particles are explicitly discouraged by the MISEV. If you see that on a product label, treat it as a red flag. In most cases, we're studying a broad population of EVs, not one specific subtype. So it's important to know what you're really getting. Everything I covered here reflects the MISEV guidelines, representing a consensus of over 99% across all experts in the field. So to summarize this video, here's the full framework to remember. Number one, extracellular particles. EPs is the overarching term, including both vesicular and non-vesicular entities. Number two, extracellular vesicles. EVs is the accurate term for lipid bilayer enclosed particles that cannot self-replicate. These are produced and secreted by all living cells. Number three, non-vesicular extracellular particles, NVEPs, are non membrane assemblies, and they co isolate with EVs more than most people realize. Number four, operational terms are permitted, but use them carefully. Your separation method influences what you're actually measuring. Number five, biogenesis terms like exosome and ectosome require demonstrated subcellular origin. In most conditions, you're studying a broad EV population, not a pure subtype. I hope this clarifies the nuances between the different terms we use in the EV field. If you found this useful, please like and share this with those who work with EVs. Follow for more.